So lactate is that byproduct of active skeletal muscle activity. So when you're running really, really hard and you're not breathing enough oxygen in, the production of lactate results. And we saw that as the pathway. And the enzyme that is involved in that is lactate dehydrogenase. So this is a source of molecules that can be used for this pathway. Amino acids which come either from the diet or from the catabolism of muscle protein can also be fed through. And we'll actually see this in future lecture. And glycerol, which is really the backbone of fatty acids. So triacylglycerol is another way of referring to a fatty acid or just a fat. These are hydrolyzed, and this is one reason that fatty acids give off so much ATP, because they have so many rounds of oxidation through the formation of this glycerol backbone. The next stage was then thinking about the nature of the reactions. And first of all, highlighting to you that glycolysis and gluconeogenesis, one is not just a reversal of the other. There are irreversible reactions. So irreversible reactions in glycolysis, so in order to make gluconeogenesis, you have to bypass them. You've got to find an alternate mechanism for gluconeogenesis to occur. And we know that both of these are important because we've already covered the importance of these pathways. So if they're both important, but we can't just turn one the opposite direction, this bypass becomes really, really important. So the first one was, remember, glucose plus ATP was giving us glucose 6-phosphate. That's one of those very, very early stages. You'll notice too that as we're going through, that we're now going into an intermediate step. So fructose 6-phosphate converting into fructose 1,6-bisphosphate, and then a late example, phosphoenolpyruvate getting converted to pyruvate. So you've got an early control point, a mid control point, and a late control point. Also, these are those reactions that are not reversible. So this allows for a better fine-tuning of the process, and you can see these are all negative with their delta G, So from an energetic perspective, these aren't going to be favourable to reverse. So that really brings us to the next question. Well, then how do we do this? What's the energy cost? We know that these are negative, so the only way you can overcome this is that you've got to add ATP or use energy. So what is the energy cost for gluconeogenesis compared to glycolysis? So we just said gluconeogenesis is energetically unfavourable unless it is coupled or linked to favourable reactions. And so what this means is that if you have a reaction that goes forward that has an excess of energy, you can then link that to a next reaction which perhaps requires energy. And by linking the two together, it allows for the net reaction to occur. So what we see is that 6 molecules of ATP, which we've seen a lot of, but also now introducing a new molecule, GTP. And the only difference is the base. Okay, these are still 3 phosphates, they still have energy, just an alternate form. These are hydrolyzed, so we take the terminal phosphate typically. to make glucose from pyruvate. But we know that only 2 molecules of ATP are generated in glycolysis, and we saw that right at the end where we were stripping off phosphate, for example, from phosphoenol pyruvate to make pyruvate. We ended up with a single ATP. So if gluconeogenesis was a simple reversal of glycolysis, it wouldn't occur because we've got 6 versus 2 ATP. We're using 6, and we're only getting 2 back. So this is not going to work. So it's that hydrolysis which allows it to occur. So as I've repeated again, nucleoside triphosphates, ATP, GTP, these are coupled to reactions to drive an energetically unfavorable reaction, and we overcome this energy defect or deficit. Now, with all these pathways, so gluconeogenesis and glycolysis, this is important, the regulation. We need to know how they occur, but we also need to know how they're regulated because they're so critical and we don't want to be wasteful, as we saw in the previous recording. So we have to have coordination between each pathway. So when one is active, the other one needs to be inactive. And that makes sense given the nature of the 2 pathways, breakdown versus formation. So both pathways are exergonic under cellular conditions. So it's therefore possible for both of them to be active at the same time. There's nothing chemically that's going to limit them from an energy perspective. So how do we control it? Again, we have allostericity. control mechanisms. So the rate of glycolysis is determined by the concentration of glucose, and the rate of gluconeogenesis is determined by the concentration of lactate and the other precursors such as fatty acids and glycerol. But if you just focus on lactate for now, this will make perfect sense. So when glucose is abundant, so remember we can't glycolysis will predominate. When glucose is low, gluconeogenesis will occur, because remember, if glucose is low, we need to make it, where this pathway comes into play. And so vice versa, glycolysis will predominate when it is high. So the key regulatory sites of gluconeogenesis in the liver is going to be that interconversion of fructose 6-phosphate, fructose 1,6-bisphosphate, and phosphoenol pyruvate and pyruvate. Those are those key control points that we have seen previously, and not surprisingly, these are the control points that allow us to control the two pathways. Now, just want to move into a disease example, and so we talk about type 2 diabetes, which everyone would have heard of, and you know that you've got the hormone insulin, and this is a signal to the presence of glucose that's in the blood. And as we just saw, when glucose is present, gluconeogenesis is not required. Why? There is no need to make glucose. If we did, again, it would be wasteful, and then we're taking metabolites or intermediates that could be used elsewhere. So how does insulin work? It turns on genes involved in glycolysis, and in turn it turns off the genes in gluconeogenesis. And this is the perfectly normal state. We saw this reciprocal regulation in the previous question. So this is the perfectly normal state. But what we see with type 2 diabetes is that even though insulin is present, Pepsi K, phosphoenol pyruvate carboxygenase, so the enzyme that's involved, it is still turned on. It hasn't been turned off. So therefore it continues to stimulate gluconeogenesis. Think back to this point here. What results is that we have now higher levels of circulating glucose. We've got glucose that has come from the diet, but we've also got this glucose that is now being made by the individual. And so this high level of glucose changes the blood osmolarity, which is what causes people to be thirsty. Because of that, there's excess urination, and then in extreme conditions, you can have blurred vision, fatigue, and headaches due to the lack of energy. So here it's a really clear example where the interbalance between glycolysis and gluconeogenesis, when it breaks down, it manifests as disease. And this is the example here. Okay, the very last question was focusing on the cori cycle. So when we think up here, we were talking about lactate, and we were saying the lactate was being produced from pyruvate when oxygen levels were low. And we saw this with active muscle cells. And you can also see it in red blood cells as well. And it can be used as an energy source for other organs. But if we focus on the muscle cell, because this is going to be the biggest producer, pyruvate levels typically exceed the rate at which the citric acid cycle can metabolize it. And so as a result, lactate dehydrogenase needs to be activated to convert pyruvate into lactate. So that is going to lead to its increase in concentration. But you can't metabolize lactate. So lactate has to be converted back to pyruvate for its metabolism. So the level rises, but then we need to shuffle it across to this intermediate. This is really the essence of the cori cycle. So in muscle, formation of lactate in the absence of oxygen allows it to make ATP, and this shifts the burden to metabolizing lactate to the liver. It goes into the circulation, goes into the liver. That conversion allows it to metabolize, and the liver restores lactate in the bloodstream back to glucose, and this in turn can be used by the muscle cells to make energy. Now the muscle cells might still take, ultimately we're going from glucose back down to pyruvate. They might still then make more lactate, but remember this is a cycle, it's a cori cycle, so this repeats, goes back to the glucose and keeps coming through. So it's a very nice way of dealing with an end product that can't be used initially, but by taking the lactate to the liver, it's converted to something which can be metabolized and in turn is going to be beneficial to the host and to the organs in this particular case. Okay, I'll stop there. As I said with the previous lecture, if you've got any questions, please get in contact.
Okay, hi everyone, welcome to lecture 11. We're moving on to glycogen, and as I've written here, how is glucose stored in the body? So study aims for this lecture are to first of all understand the general structure of glycogen. It's different to what you might have seen previously. We'll talk about the different roles of glycogen in muscle and liver, because these are the two main locations where it's going to be present and also it's going to be used. We'll then move on to talk about how glycogen breakdown is regulated in response to key hormones, and these ones will be ones that you've heard of before, and also we'll then finish off by talking about hormone-driven regulation of glycogen mobilization, which those two are very much related. So as always, you've got your section here for questions. You've got reference to the textbook as well. Now, glycogen is a storage form of glucose, and so this is in animals. If we talk about storage form of glucose, say in other organisms like plants, it's going to be starch. So they're just as an example of the two different types. But what glycogen is a polymer of glucose. So this is where you have a string of glucose molecules that are joined together, and they're mostly, not completely, but mostly covalently linked by what are called alpha-1,4 glycosidic linkages. So glycogen, glycosidic, that's the link between the two. So when you see glycosidic, think of glycogen. And the 1,4 is really telling you the position of the bonds. Why is it not all of them? In the sense that I've got here mostly, because it has a branched structure. So glycogen is not a linear molecule, it has a branched structure, as we'll see in the diagram below. So every 12 residues, there is a different bond, and it's called an alpha-1,6 glycosidic bond. And in order to create this branching structure, you have to have a different type of bond, and that's why we've got this. So the vast majority is 1,4, but you've got occasionally this 1,6 bond. Now, why do we not use glycogen as our main energy store? It's because it has a variety of reasons, but the first one is a limited reduction. So remember, reduction here is talking about redox and oxidation, so it has limited rounds of reduction. It doesn't yield as much ATP as the complete reduction of fatty acids, and we'll talk about that in the fatty acid lecture. But fatty acids can go through many more rounds to generate ATP. But it is readily metabolized, and so this is a ready source. It's an easy, quick source, whereas fatty acids are more of a longer term source of energy. And it can be used in anaerobic conditions. And that's important because under exercise, where oxygen levels might be reduced, you need to have an energy source. And for fatty acids, that's not going to be possible. So we use glycogen. And because of this reduction limit in ATP, we just don't have the store. So you can see, as we'll get into later, we just don't have the same amount as we might have for fatty acids. Now, as we saw with the learning objectives, we're going to talk about liver and muscle. And the reason for that is that they're the major stores of glycogen. So liver and muscle is the major location where we'll find glycogen. And again, this is either to control the level of glucose within the blood, so for the liver, and in the muscle, it's really for that activity. And it constitutes 10% of the weight of the liver versus only 2% by weight in the muscle. So you can see you've got a very limited supply in the muscle. This is going to be expended very quickly with exercise, and then you need to use alternate sources. And as I mentioned before, the liver serves to maintain blood glucose levels so that you have a homeostatic control. And skeletal muscle, the glycogen there is to serve the energy demands of muscle on demand. And so this is when you've got a fight or flight response, as an example, where you have to run, then you need rapid energy. Now, in terms of the storage, glycogen exists as small granules within the cytoplasm. The size doesn't really matter, 10 to 40 nanometers, but very small. And they each contain about 55,000 glucose molecules. So it's very, very densely packed, and that's an important concept. And this dense packing really comes about with those bonds. I've highlighted this again, we've mentioned it before, but whenever you see something in bold, just to highlight the importance, in the liver, glycogen synthesis and degradation are regulated to maintain blood glucose levels. So this is a key theme that we will talk about. The level of glycogen within the liver. If you've got too much glucose, you're going to try and build up more glycogen within limits. And if there is a demand for glucose, this is going to be a quick and rapid release mechanism, so the degradation of glycogen to release the glucose, which can then be used for energy. So in this diagram here, this is showing first of all the branch structure, so a very gross overview of glycogen. Now here you've got a letter G, so as it says here, a protein called glycogen. This is important for the formation or the initiation of this cluster. You see you only have the one protein and then you have all of the other bonds. Remember that most of these that are linear are alpha 1, 4. These points of junctions where they branch off are alpha 1, 6 bonds. So structure of glycogen now shown here. So each one of these individual units is glucose, but because this is a polymer, these have been joined together, and you can see that the most common bond is going to be the 1, 4, and we see it here. So one position on this glucose molecule joined by an oxygen atom to the 4th position, and if we go to the 6th position and the 1, 6 across here. So this is where the branching occurs, and this is the linear profile. You can see here the two ends, the non-reducing ends. This is really just about the points of reactivity, but this is going to go from left to right and it can extend. Now, glycogen degradation. We mentioned up here the liver, glycogen synthesis, so making it, but degradation as well, maintains blood glucose levels. So what is glycogen degradation? It's glycogen lysis or glycogenolysis. So whenever you see lysis, it's cutting, and we'll talk about it multiple times, but we will talk about lytic events or lysis, particularly with fatty acids, because with fatty acids we have to chop them off 2 carbons at a time, so this is where you'll see it a lot, but it's the same idea here, glycogenolysis, where you're starting to break the glycogen, into its glycogen or glucose units. So breakdown of glycogen yields glucose 6-phosphate. So not glucose on its own, but glucose 6-phosphate, and this is an important addition for activity. Now you might think, well, okay, we just need to cut the bond and off we go. Actually requires 4 enzymes. It's a more complicated process. The first one is to degrade the glycogen, which is this first part. The other 2 to maintain it as a substrate, because it needs to be metabolized, and then 1 to convert it into that suitable form for metabolism. And this last point is also very important. Whatever your precursor molecule is, it needs to be in a state that is able to enter the metabolic pathways, so that whatever energy, whatever ATP it's able to release is in a form that is compatible. If it's not, then it can't enter those pathways. You're not going to generate that ATP. So very, very important concept. So let's go through these. So the first one to degrade glycogen. So we have a phosphorylase, cleaves glycogen to release glucose 1-phosphate. So this is the enzyme that is cutting or leading to that lytic event. So glycogen phosphorylase is the key regulatory enzyme of glycogen breakdown. As I've mentioned in previous lectures, whenever we talk about pathways, those key molecules, the rate limiting step, the initiating molecules, the initiating enzymes are critical to your understanding. So this is one that you must know, because really it is the first one that allows you to go from glycogen to glucose, and it also is the key regulatory point, so it controls all of those downstream effects. So this is a really, really important enzyme. So it cleaves glycogen at that 1,4 position. Could have put alpha, but we know it's the 1,4 position. And it does this by adding orthophosphate. Now orthophosphate is actually 2 phosphates, and this breaks the bond to orthophosphate is 1. I was thinking of pyrophosphate. So orthophosphate is 1 phosphate, and that leads to glucose 1-phosphate. Look, it's on a different position because we want a glucose 6-phosphate. So make sure you understand the difference here. This is just to release it, but remember it's not in the state that we wanted, which is why this last enzyme at the bottom here is required. So glycogen plus phosphate, inorganic phosphate. We have lots of phosphate within the body. You know that ATP has 3 phosphate molecules, so there's a ready source available. This converts or splits one of those glucose molecules to glucose 1-phosphate. We've now got the glycogen that continues, but we say N-1 residues because we have lost our residue. We have lost 1 glucose molecule, which is shown here. And this process is called phosphorylysis, phosphorylysis, because we've got a phosphate group and we're lysing. So it's the same idea, it's just adding the phosphate as the molecule, which is breaking the bond and releasing that particular glucose molecule. So the phosphorylase, which we've got up here, glycogen phosphorylase, leads to the sequential removal of glycosol residues. Glycosol is the correct term, but you could also just say glucose residues, because remember, it's just a string of glucose molecules. These are removed from the non-reducing ends of glycogen. So these are those ends with a free hydroxyl group. So here is a glycogen molecule. You can see here we've got a free hydroxyl group. Here there is no free hydroxyl group because there's going to be a bond. So this is the free one with N residues. So here it's 4. Here's the phosphate coming in. Obviously the enzyme is going to be acting. We add it to this bond here to break the alpha-1,4 glycosidic bond, and we see the release of glucose 1-phosphate, because remember this is 1 to 4, so this is the 1 position, and we then have the glycogen residue which has been released, and it's N-1 because we've lost this particular glucose. If the cell needs more, what does it do? It just keeps going, chop, 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 keeps going until it needs to remove that molecule. Now, glucose 1-phosphate, we don't want that, but not surprisingly, it's readily converted to glucose 6-phosphate. Makes it very easy for the cell to use. This is the preferred and the required substrate. It's energetically favorable because we have this phosphate group. And what you can think of here is this is very similar to what we see with ATP. When we talk about energy within the cell, we talk about ATP. Where is the energy? The energy is in the bond with the phosphate, and releasing that phosphate releases energy. And in essence, we've got the same thing here. We've got a phosphate group attached to the glucose. Releasing it, moving it, because we're going to have to move it to a different carbon, is going to release energy, which makes this thermodynamically or energetically favorable because of that phosphorylation state. So it's going to facilitate this process. So one step facilitates the subsequent step. What else do we need? We need a debranching enzyme, and this is required for glycogen breakdown. Remember that glycogen is a highly branched molecule. So 1 in 10 residues of glycogen are branched. Glycogen phosphorylase cannot cleave at the alpha-1,6 bonding glycogen. So recall before we said the phosphorylysis occurred at the alpha-1,6 bond, alpha-1,4 bond, but we have alpha-1,6 bonds also being present. So how do we work at that particular location? So what we see is that the glycogen phosphorylase actually stops lysing at 4 residues away from that branch point. So if we say that there's 10 residues, well, 4 away means that only 6 molecules of glucose would be released. It's a great reduction in the total capacity that could be released, so there has to be another way. And what we see is that the remainder of the glycogen is degraded through a process called remodeling. And this is basically just shifting the groups around so that the enzyme is able to continue its activity. So remodeling an activity of a transferase. And the transferase, as its name suggests, transfers groups. And there is also an alpha-1,6 glucosidase. So these are the 2 enzymes that are important in this remodeling process. What does it do? The transferase shifts a block of 3 glucose residues, remember, glucose, interchangeable term, from one outer branch to another. It removes those last 3 and shifts them elsewhere. And by removing them, there's now a continuation of that linearity, and the enzyme is able to continue to release those individual glucose molecules as is required. And so what this does is that it exposes a single glucose residue which is linked by an alpha-1,6 linkage. That should be alpha, not A. Apologies. And this is hydrolyzed by the alpha-1,6 glucosidase. So remember, we had the second enzyme that is able to cut at that location. And that free glucose is now phosphorylated by another enzyme which is called hexokinase. You can tell what this is going to do. Hexo is telling you 6. Glucose is a 6-carbon molecule. It adds phosphate. A kinase is an enzyme that will add phosphate. So 6-carbon phosphate adding molecule. And in this context, we know that that substrate is going to be glucose. So if we look at this diagram, here is a string of glycogen. So we've got our alpha 1,4 linkages in a line, and it's saying core because this is where it's linked to the protein, and this is the first branching point. And then we've got another string of alpha 1,4, and here's the alpha 1,6 linkage. You want to remove those glucose molecules. We've got lots to cut off. And so here we're showing 8 that are being removed. So there's 8 phosphate molecules, just showing you as a collective. You could repeat this 8 times, obviously, but it's just showing you as one. And this is where the phosphorylase comes in. We now have a branching point, and we've got 4 glucose molecules. And we said, well, when we've got 4, phosphorylase is not able to work. So what happens? Transferase comes in, it cuts this bond, and it shifts it to the end of the previous grouping. And so if we see in this diagram, these 3 are now attached to the yellow glucose molecules here. It's taken it and physically moved it across. And so we've now got 1 glucose molecule here. Now this could be problematic, but remember, we've got an alpha 1,6 glucosidase. It acts on this, chops it, and removes it, and we now have this string of linear alpha 1,4 glucose molecules. Phosphorylase is able to come along and just cut, and we've no longer got that issue. So it does require some additional steps, but you can see limited requirement in terms of activity. By removing that group, you've got this massive linear glucose chain, which allows for the enzyme to be active and in turn release all of those molecules. So as it says, diagram showing the glycogen remodeling, action of the transferase, removing 3 glucosyl units, so 3 glucose molecules in blue, 1,6 glucosidase which hydrolyzes, so hydro referring to water. You can see why I've got water molecule here to remove that last glucose molecule. This diagram is now just showing you that hydrolysis. We've shown it simply here, but here is another way of demonstrating this. So here's glycogen, we've got that bond. This is the 1,6 bond because this is what we're looking at here. You've got the water molecule, you've got the alpha 1,6 glucosidase. Based on the nomenclature here, we know that this is a 1,6 bond because this enzyme would only work on a 1,6 substrate. This gets broken, and you can see that what you're going to do is that oxygen is going to be converted to a hydroxyl, and you've got the same thing occurring on this side here. So we've got glucose and the glycogen residue as well, but it's now shortened by 1 glucose molecule, and this can then go off and be used. What else do we need? The third one, phosphoglucomutase. Phospho for the phosphate, gluco for the glucose, and mutase. Mutase can be thought of as a converter. It converts it from one state to another. That's all it is. So this is taking glucose with a phosphate group and converting it from one form, so glucose 1-phosphate, which is what we had released initially, and converting it to glucose 6-phosphate. In essence, is shifting the position of the glucose from the 1 position to the 6 position. That's all. As I've written here on the second line, a mutase is an enzyme that shifts the position of a phosphate group on a substrate, and it contains a phosphate group itself. So the enzyme contains a phosphate group on a serine residue, and the serine residue is part of the active site. Now, why is this important? This phosphate group on the serine is involved in an exchange reaction. So it is exchanged to the glucose 1-phosphate, and the 1-phosphate is then removed. And in turn, it's returned back to the enzyme, and this now leaves the glucose as glucose 6-phosphate. It's essentially swapped over. The phosphate that is on the mutase enzyme is transferred. The 1 position is removed, given back to the enzyme, but is now transferred onto the 6th position. So it's a bit of a rearrangement. And by transferring the phosphate back to the phosphoglucomutase, it retains its activity. It's able to repeat the next cycle. We've been able to shift the phosphate from the 1 position to the 6th position and now convert the glucose into a substrate which can be used metabolically, which can be used for enzyme action and of course for energy production. So this is what this is showing. What we can see down here on the bottom, we're in yellow, this is the phosphoglucomutase. It's just showing you a very small portion of the active site. And you know that the active site is going to be a protein, and proteins are made of amino acids. And here we've got serine as the amino acid with the phosphate group attached. Here is our glucose 1-phosphate coming in as the substrate. and here is the phosphate on the incorrect position. Incorrect for metabolism. You've got glucose 1,6-bisphosphate, so this phosphate gets attached to this group, so that's the first step. So the mutase donates it. It's 1,6-bisphosphate because we've got 1, 2, so biphosphate, and they're on the 1 and 6 position. This is no longer required, so it gets donated back to the mutase, and we can see it's now brought back to this location, and in doing so, we've been able to convert the enzyme back to its preferred state, glucose 6-phosphate state, which is shown here. This is now able to go into metabolism and can be readily used. So I'm going to stop there simply because I think it shows really nicely how glucose can be released from glycogen, the importance of the first step, certainly the importance of these intermediate enzymes.
Okay, so we've released the glycogen, so now we've got to talk about the release of glucose from the liver. And as we've said now multiple times, major function of the liver is to maintain a constant level of glucose in the bloodstream. And this is important for homeostasis. The phosphorylated form of glucose is not transported out of the cells. The phosphate interferes, probably through a charge interaction with the transporter, and that interferes with its ability to be released. Now, we know that the liver contains the enzyme glucose 6-phosphatase, and we saw this in the prior lecture with gluconeogenesis. And this is a hydrolytic enzyme. And what it does is that it removes the phosphate group from the glucose 6-phosphate. So you can see that here, very simple reaction, glucose 6-phosphate, which is what we had at the beginning, through a hydrolysis gives us glucose and the free phosphate. That glucose can now leave the liver and enter the bloodstream. So we've got storage of glycogen, and we said that this is mostly in the liver. We then have glucose 6-phosphate being produced, but it can't get out into the blood. And so what we now do is we remove this group, and this is now able to exit and go into the blood. That's all that we're talking about. It's a very simple sort of concept. Now let's talk about the regulation process, because the release so far I think has been fairly simple and straightforward. So let's think about the phosphorylase activity. So glycogen degradation is controlled by several mechanisms, and these all focus on glycogen phosphorylase. So this is going to be the key enzyme that we need to focus on. It is controlled by several allosteric effectors, and these all signal the energy state of the cell. And what we mean by this is if the energy is low, energy needs to be replenished, and so we want the glucose to be released, so ultimately we get ATP. If ATP levels are high, the drive to make additional ATP is not there, so the reverse would be true, that we don't want the breakdown of glucose, and therefore we don't want the breakdown of glycogen. So that balance is really quite important. So we've got these allosteric effectors. We've also got the ability for reversible phosphorylation. And remember, this just means that we can add the phosphate and we can take it away. And this response is mediated by different hormones. And you would have probably heard of most, if not all of these. Glucagon is one, adrenaline you would have heard of, and I know insulin because we spoke about this in the previous lecture. Now we're going to compare the control in the liver and the muscle, but we will do this separately. So make sure you understand what happens in the liver, and then when you get to the muscle, you can then compare and contrast the differences between the two. That will make the learning an easier process for you. So, in the liver, the enzyme, and what are we talking about? We're talking about the glycogen phosphorylase, as it says up there, exists in two forms. There's an active phosphorylated form, so phosphorylase A, so A equals active, and there's an inactive unphosphorylated form, the phosphorylase B. Now, B doesn't lend itself to anything that tells you that it is off. So if you remember that A means active, then you know that B means off. And as we saw previously, we said that the active state refers to the R state and the less active state is the T state. So in the liver, it exists mostly in the active A form, so phosphorylase A or the R state. Don't get caught up in this terminology. We're talking about the same thing. And to be honest, if you just remember phosphorylase A and you don't worry too much of the R state, I think I would be happy with that. So by doing so, we're going to get the conversion, we're going to make glucose, and this is going to provide glucose to the whole body. The binding of glucose to the enzyme changes it to the off state. So glucose is itself an allosteric modulator. Okay? The substrate can modulate the activity of the enzyme. And by doing so, it makes it less active because excess glucose must be present if this is occurring. And so therefore, there's no need to degrade the glycogen. So this is a nice control mechanism that takes place. In contrast, if you look at the muscle, what we see is that in muscle, the default state is actually the inactive B form of the T state. So this is the off state. And this makes sense because in muscle, you don't want to be breaking down glycogen if the muscle is resting. It's only when it is active do you want it to do that. So only activated during muscle contraction. And this is where you'll see the shift between the B and the A form. It's also activated by high levels of AMP. And again, remember what we said, ATP goes to AMP and phosphate or phosphate itself. So AMP, if this is high, it means that the level of ATP is low. So more energy is required. And it makes sense then that we would want to activate this pathway. And similarly, where we said glucose can block it, here we can see that glucose 6-phosphate can actually stabilize the B form. It will turn it off by keeping it in the off state, and this prevents excessive breakdown. So phosphorylation converts phosphorylase B to the A form, and it occurs at a specific serine residue, and it is initiated by various hormones within the body. So low blood glucose leads to glucagon expression. Exercise releases adrenaline. So we've got the two regulatory molecules that are occurring. So these lead to phosphorylation and hence activation of the enzyme. Now, how does this signal get received for glycogen breakdown? Now there should be from, not form, so from glycogen breakdown. There is a kinase called protein kinase A, PKA, and we'll talk about this in more depth later in a subsequent lecture on signal transduction. But all you need to know is that this is a particular kinase that is very important. And it is the one that is responsible for phosphorylating the phosphorylase. So again, the B going to the A form, because remember, this is the off state and this is the on state. So we want to add the phosphate. So what is the cascade for this response? Now, let's have a look first of all at the hormonal process. This is one of the diagrams that we haven't yet covered. So diagram showing the hormonal control of glycogen breakdown. If we look at the diagram, there is a left side and there is also a right side down here. So the left side shows the hormonal response to fasting, as we see here, so low glucose. So glucagon stimulates glycogen breakdown in the liver. Glucagon will want to stimulate the breakdown of glycogen because glucose is low and we want to increase it. So what we can see here, glucagon is released from the pancreas. In the liver, glycogen is broken down to glucose and it enters the blood. Now don't forget, particularly when we come to talk about it on this side, the cori cycle. Lactate can be converted to glucose and that glucose can then go back into the muscle. Remember in the previous lecture we spoke about lactate being produced, a bit of a dead end, we called it, metabolically. It goes into the liver converts it back to glucose, and then it can go back into the bloodstream and make its way back to the muscle. So these are all very much interconnected. On the right-hand side, it shows you the hormone response in response to exercise. So this is vigorous exercise. We have adrenaline being produced. So this is an American textbook or North American textbook, so it's referred to as epinephrine here. We would say adrenaline, same thing, just a different name. This is reduced from the adrenal medulla on the kidneys, goes into the bloodstream. And you can see here epinephrine comes across, binds to the transporters that facilitate this process, so glucose coming into the bloodstream. So that enhances glycogen breakdown in the muscle and the liver to provide fuel for exercise. So again, if you look here in the muscle, glucose is broken down to glucose 6-phosphate, pyruvate, and lactate, because this is an energy-intensive process. The glycogen that is within the muscle can also be converted, and this in turn gives rise to that energy that is occurring. And remember, the lactate goes back into the blood. Now with this diagram, they're showing it going down, but really it's going down, it's going to come back up through the top and then go back into the liver. And you can see that with this diagram in terms of how it's mixing together. So this is linking in what we've seen previously, but now just showing how glucagon is affecting glycogen metabolism. Now, if we look at the signal transduction pathway, so diagram showing the signal transduction cascade activating the phosphorylase enzyme. So remember, B goes to A. So the hormones, glucagon and adrenaline, we've seen them in the previous diagram. How do they work? They activate a receptor. So you know that a hormone will bind to a receptor. That receptor leads to an increase in a molecule called cyclic AMP. So AMP, ATP can be converted to AMP, but then if you've got a bond that makes it more of a circular molecule, it's referred to as cyclic AMP. Cells are very sensitive to the level of cyclic AMP, particularly when it goes up. And this increase in cyclic AMP is what activates the protein kinase A. So the A in the cyclic AMP refers to the A in the protein kinase. So you can see when this goes up, this is going to become activated. So as it says here, this activates pKa, which activates the phosphoryl kinase. It phosphorylates the B form to the A form, and we start to get conversion. Now the structure of the molecule up here, so here is the receptor, epinephrine and glucagon. They bind to a receptor. The 7-transmembrane receptor will be covered in depth in the signal transduction lecture, so don't worry too much about it. But it has these subunits that essentially, when they get activated, act on this enzyme, which is called adenylate cyclase. And adenylate cyclase is the enzyme responsible for converting ATP to cyclic AMP. And as we said here on the right-hand side, protein kinase A is very sensitive to cyclic AMP, and the red is shown as the off, the green is shown as the on state. So that binding turns it on. The protein kinase now activates the phosphorylase, and the phosphorylase then acts on the enzyme to go from the off state to the on state and allows that process to occur. So this demonstrates the cascade and also demonstrates the linkage of activation of hormones and the transduction of signal And so when we look at glycogen degradation and synthesis, this is no different. It's not just the reverse. It's a little bit different. So glycogen synthesis uses a molecule called uridine diphosphate glucose, or UDP glucose. And uridine, again, we'll see this when we come to talk about nucleotides. So again, this is another example where we've got a molecule that we think about in one context, but it's clear has a function in different contexts within the cell. And so this is used as opposed to glucose 1-phosphate. And it uses the UDP glucose as the activated glucose donor. Remember, activated means phosphate. It's got a phosphate molecule, and it is carrying a glucose molecule to donate. And that donation allows for the glycogen to be synthesized. That's all it's referring to. It is just a molecule that is carrying a phosphate and a glucose, just a different molecule. So here we can see in synthesis, what do we have? We've got glycogen, which has a certain number of units, because remember it's a polymer, and we add the UDP glucose, and this is going to lead to that glucose being deposited onto the glycogen. You can see we've got N+1, and we release the UDP molecule, the uridine diphosphate. The reverse, the degradation, is as you would expect. Glycogen, which has got some residues, the empty UDP and phosphate, and we strip off one of those glucose molecules, and we generate glucose 1-phosphate. And remember, the glucose 1-phosphate has to be then converted to glucose 6-phosphate. So it's just another way of this occurring. But we're not interested in this side, we're more interested in the synthetic side. And as I mentioned before, UDP glucose is the activated form of glucose, so it facilitates the process. And if you look at the diagram, you can see here's the structure. The glucose is shown in blue, so I'll just highlight that for you here. And we've got 2 phosphate groups, so one here and the second here. And this is then the uridine group down here. And remember, it says here, the 2 phosphate groups which provide the activated nature of the molecule. There is inherent energy in this process and a point of reactivity. Now, generation of UDP glucose is shown in the next figure. So here is glucose 1-phosphate. Here is uridine triphosphate. Remember, we've got 3 phosphates. We're going to lose one of those phosphates, which is why we then go to the uridine diphosphate version. So remember, nucleotide base with the phosphates. These two get joined together, and here is our glucose and the 2 phosphates linked to the uridine, and here we've got pyrophosphate, which is just 2 phosphates that have been released. We can see that here that we've lost those 2 phosphates. This one and this one has now been removed because we've joined this one and this group. So we cut off at that location and we form the bond that is taking place. Now, glycogen synthase. So we know that glucose makes up the glycogen. So this is the glucosyl units. These are added to the non-reducing end of glycogen, and we spoke about this previously in relation to the hydroxyl group. The enzyme that makes it is glycogen synthase. Just as we would say, when something is synthesized equals its production. So glycogen synthase synthesizes glycogen from glucose subunits. And as a result, it is going to be the key regulatory enzyme of this process. So if I was to ask you a question about glycogen synthesis, you would have to be thinking about glycogen synthase as that key point. What does this enzyme do? It's responsible for the transfer of glucose from UDP glucose, which was the activated carrier that we just spoke about, to the growing chain of glycogen. It's what takes this glucose and just adds it to what is already existing. And not surprisingly, there are two forms, one in the liver and one in the muscle. And what we find is that the synthase only adds glucosyl residues to a polysaccharide with 4 or more residues. And as a result, what is called a primer is required. So if we had 1, 2, or 3 glucose molecules that are joined, so a small glycogen molecule, the next one, so number 4, wouldn't take place because it needs at least 4. And that's probably part of the binding and some of the stereotactic interaction that is required. So what we say here is a primer is something that facilitates this shape and this structure and allows it to occur. And in this case, we will see a molecule called glycogen, and that is what is involved. That allows for it to occur. Now, if we go back to figure 11.1, I didn't cover the structure at the beginning because it wasn't really relevant at the point in time. But here we have the glycogen molecule. We can see all the bonds and the linkages. But look here. This is the glycogen. This was the initial primer. So each one of these containers, or each one of these glycogen granules, is going to have one of these glycogen in the middle, or at least as the initiator for this entire process. And now you know why that is the case. We'll just go back. Okay, so that's where we're finishing off. Now, I might just stop there before we start to move on to the next part, and also not to make this recording too long. So, see you. Okay, welcome back. So we now want to move on to talk about the alpha-1,6 linkages. So we know that glycogen synthase, as we've just seen in the previous section, adds to glycogen, so it's got 4 residues, adds to the glycogen and builds glucose at a time. But it only makes the alpha-1,6 linkages. Now, we know this will give us a linear chain because that's what we've seen in the diagram. But we know that glycogen is branched. So therefore, we need a branching enzyme to introduce this other linkage, this alpha-1,6 linkage. And what it does is that it transfers 7 residues to create the new branch. We'll get to the diagram shortly. But why do we have this branching? What is the point of this? It increases the solubility of the glycogen, and obviously this is going to be important. If it's insoluble, it's not going to be very easy to store. It's not going to be very easy to access. So by having it in a stable or soluble form, it's much more readily accessible. It does affect the density because of that branching nature. It also does affect the degree of reduction, as we said, and as a result, there's going to be less ATP generated. But remember, this is not meant to be a long-term energy storage. That's where fat, in principle, comes into play. It also, by having this branching, also accelerates the rate of synthesis and degradation because it's easy to attack multiple sites, and so you can actually then cleave it at multiple points. So what do I mean? Well, if we had a linear glycogen molecule and we could only chop at one end, we would have to wait until each reaction occurred. But if we've got a branching point, okay, something like this, probably not 100% correct in my drawing, but that's okay, you get the point. You can see that you can start to attack at multiple points you can accelerate either the cutting or the formation as it's occurring. So it accelerates, there's multiple points of attack that occur within the actual molecule itself. Now, glycogen synthase, it also exists in an active, so A, non-phosphorylated form, and an inactive B, phosphorylated form. This should make you feel a little bit more comfortable because this is now some repetition. So once you understand the process of one, A for active, and this is for the phosphorylases where we spoke about that, but now we're talking about the glycogen synthase, it gives you the same idea. Now, what activates the glycogen synthase? Well, as you would expect, glycogen synthase kinase, so GSK, glycogen synthase kinase. It phosphorylates the synthase in response to insulin and protein kinase A. So the control of glycogen synthase and the phosphorylase are opposites. And that is important because remember, phosphorylase was release, the synthase is production. So you would expect them to be opposing reactions, opposing processes. So again, I've put the asterisks here because this is another example of somewhere where you need to know the information on control. So this figure here is showing you the branching that leads to the formation of the alpha-1,6 linkages. So here is the core, the glycogenin is the starting point. Here we've got those alpha-4,1 linkages, and you remember that these are linear. We've got the UDP glucose, which is our substrate, because this is what brings in the glucose. And we've got our enzyme, which is the glucose synthase, which is just going to add it on and on and on. And you can see we've got 1, 2, 3, 4, 5, 6, 7 molecules of glucose that have been attached. We're getting to the point where we're going to have to branch. And so what we now see is that that group is taken and is moved, and we form this alpha-1,6 linkage. And that branching now gives us 1 and 2 points of attack, which is really the diagram that I was showing you here on this side, but from the opposite direction. And remember, that accelerates the rate of synthesis or degradation depending on the direction that you're going. So extends both non-reducing ends followed by more branching. Now, what about the regulation of glycogen breakdown and synthesis, so both components? As we saw with glycolysis and gluconeogenesis, opposing pathways have to be tightly controlled. And as we see here, glycogen is prevented from being synthesized while it is also being degraded. That would be a wasteful process. So the same pathway that activates synthesis turns off degradation, and we'll see that in this diagram here. So phosphorylation of glycogen synthase kinase by pKa turns off its activity. So as I mentioned, this is the reversal of the process that is achieved by a phosphatase. So this is a unique setting. Typically we see phosphorylation being associated with the on state. Here it is the opposing effect. So if we look here, coordinated control of glycogen metabolism, activation of pKa stimulates glycogen degradation and prevents synthesis from occurring. So we've seen this before. We've got enzymes, hormones binding glucagon or adrenaline, binding to the receptor, activating the cyclase, cyclic AMP goes up, and protein kinase A becomes activated. And we saw that it activated the kinase, which activated the phosphorylase, and this in turn led to the release of glucose 1-phosphate from the glycogen. So this is the breakdown. What we can see here is that protein kinase A interacts with glucose synthase kinase, and by doing so, it phosphorylates the synthase and converts it to the off state. So you can see here that this pathway is stimulating release. This pathway, which is the opposing effect, is going to now be blocked. We've turned off the synthase enzyme so that we've got this reciprocal control. So really quite an important mechanism. And if we now look here, we want to talk about the phosphatase, because remember, this is now different. What we were talking about before was typically the phosphorylation. So now the phosphatase is going to be important. So we've got the kinase, which is on, goes to the off state, which is the phosphorylase kinase, and the off, remember, red is off. And here we've got the inhibition. This is all mediated by our phosphatase at the top. Now the 1 is just really the name of the phosphatase, but I'm not too bothered about you knowing the name. Just know that it's a phosphatase, and it's able to inhibit these steps. The phosphatase stimulates the synthesis of glycogen, so it's blocking on this side, and it is turning on this side. It's stimulating glycogen synthase B. Remember, the synthesis of glycogen, the off to the on state. It takes the glycogen, it takes the UDP glucose in the activated form, and it extends it by 1 residue. This is really no different to what we've seen above. It's just now including the phosphatase. It's putting the phosphatase into the picture and explaining it in more detail. Okay, so what about insulin? We've spoken about glucagon and adrenaline. What about insulin? We know that when blood glucose levels are high, insulin stimulates glycogen production because we want to store it. But the question again is, well, how does it do it? How does the cell do this? Insulin inactivates glycogen synthase kinase. And notice, when we have a net result, an end result that either we want storage or release, the hormones pinpoint the key molecule in the pathway. So we inhibit the synthase kinase. Insulin receptor is a tyrosine kinase. By binding, it activates the kinase. So all this is telling you is that the receptor is a kinase. We don't have to recruit, so there is no recruitment of a kinase. It has its own inherent activity. By doing so, it phosphorylates the receptor, and it phosphorylates substrates, which are called insulin receptor substrates, IRS. These phosphorylated proteins lead to the recruitment of another protein, which is called glucose transporter. And this allows the glucose to enter the cell. The activated protein kinases phosphorylate the glycogen synthase kinase, and the opposing effect is going to the protein phosphatase 1. So protein phosphatase 1, PP1, dephosphorylates the glycogen synthase to turn off the cycle. So this is where we can see insulin binds to the receptor has intrinsic kinase activity, and we can see here we've got this insulin protein that gets phosphorylated, which amplifies the reaction on the kinase. The synthase kinase gets converted from the on state to the off state, and by doing so, it interferes with the glycogen synthase production. So you can see that you can have this loop that is occurring based on the insulin production within the cell. Okay, let me just finish there. We've got the last part. What is the effect of glycogen metabolism on blood glucose levels? We've spoken about this multiple times. The liver maintains blood glucose levels fairly tightly controlled between 4.4 and 6.7 millimolars. As the level of blood glucose rises, phosphorylase A, which is the active form, decreases. Remember, this is what is making glucose. So as glucose is going up, we want to turn this off, while the synthase activity starts to increase because we've got this excess substrate. So this diagram here is just showing you, if you were to take a glucose meal, what would happen to the activity of these enzymes. So we've got enzyme activity, depending on which one it is, over time. So if you look at the phosphorylase A, at low levels it's going to be active. As glucose is added at time 0, you can see that it starts to drop and it gets down to a resting state, which is now in the off state. Correspondingly, the synthase, which is off because there is this massive difference between the two, this is the off state. Over time, there's a crossover point, it starts to get activated, and it now goes on to the on state and is able to take that and store the glucose away as glycogen. So again, very clever control mechanisms, but you can see the importance of being able to turn things on and off to store and to release the glucose within the body and within the cells. And remember, our focus has always been predominantly on the liver, but we've also spoken about the muscle. Okay, you've got your summary points here. Make sure you go through those, make your own notes about the key control points, and you've also got your tutorial questions, so make sure you do those to help your understanding. You can see there's a few. Hi everyone, this is a re-recording of the glycogen tutorial. I'm not sure what happened with the previous recording, but that's okay. So we'll go through the questions, explain the structure of glycogen, highlighting the types of glycosidic linkages, what's the significance of the branch structures in glycogen molecules. So, you would know that glycogen is the polymeric storage form of glucose, and it is mostly alpha-1,4 glycosidic linkages. This is the most common. There's a branch structure, and so every 10 to 12 residues, you have an alpha-1,6 glycosidic bond, and this obviously having this additional bond allows it to branch out. The benefits of branching are the following. First of all, it increases solubility in water, and you know that cells are predominantly water, so that's an important aspect to keep it within the cell. Having the branch structure also facilitates mobilization, but rapid mobilization. So the enzyme glycogen phosphorylase can act simultaneously at multiple branch points, and so it can break down the glycogen into glucose very
and that facilitates that breakdown really quickly. It also makes it more compact. So without branching, if you had a linear glucose polymer, as you see in starch, it's much less efficient. So the branching actually gives it some benefits in the packaging. And from regulation, glycogen synthase and glycogen branching enzymes control the addition and the positioning of the branches during synthesis. So this makes sure that glycogen is synthesized and controlled and degraded in a controlled manner. So there's more of a regulatory. Perspective. So really, quite a few different aspects that relate to glycogen and its metabolism. So then we moved on to talk about the role of glycogen in really liver and muscle. These were the two that we were always comparing and contrasting. So compare in the storage and function of glycogen in the liver and skeletal muscle. Why is the liver crucial for maintaining blood glucose levels? And how does skeletal muscle utilize glycogen? So, as we said, the major stores of glycogen are the liver and the muscle. If we start with just the liver, 10% by weight in the liver is glycogen, whereas 2% by weight in the muscle. Muscle is actually higher, but you've got so much more muscle compared to the liver, it's actually a lower amount. And the liver serves to maintain blood glucose levels. It is the homeostatic level of glucose within the blood, therefore facilitating glucose throughout the entire body. In contrast, skeletal muscle, the glycogen that's in the muscle there is simply to serve the energy needs of the muscle on demand. So as the muscle, if you have to go for a quick run, the glycogen that is there is going to be used as the energy source. The skeletal muscle does not release the glucose into the bloodstream, rather the glucose is used locally within the muscle for that energy production during contraction. So if it's not used, it will get converted back. It doesn't go back into the circulatory. respiratory system, so quite a different end use. Now, in order to access the glycogen, we have to talk about the enzymatic processes of glycogen breakdown. And so, as it says, specifically focusing on the role of phosphorylase, because this is the key enzyme. How does phosphorylase glycogen and what is the significance of phosphorylasis. So remember, breakdown of glycogen gives rise to glucose 6-phosphate, and there are 4 enzymes that we'll talk about. So first of all was the phosphorylase, where we got cleavage of glycogen to release glucose 1-phosphate. The glycogen phosphorylase is that key regulatory enzyme, as we mentioned before. So it's the first step. It's going to be the rate limiting step. We've seen this time and time again. It cleaves glycogen at the alpha-1,4 position, and it does so by adding orthophosphate to give rise to this glucose 1-phosphate. And as we mentioned above, this is the whole process of cleaving the glycogen is called phosphorylasis. So remember, phospho refers to the orthophosphate that has been added, and we're breaking it, so this is the lysis. Glucose 1-phosphate is then readily converted to glucose 6-phosphate. It's just a positional thing, so in terms of where it goes. We then have the debranching enzyme, and I was going to talk about it, but it actually is in part of the next question, so we'll talk about the activities of the debranching enzyme soon. We've got the phosphoglucomutase. So when you see the mutase, remember this is just converting from one location to another. So in this case, converts glucose 1-phosphate to glucose 6-phosphate. Shifts the position of the phosphate group on the substrate. Now, phosphogluco, well, it's glucose. It's got a phosphate group attached, as we see here, but it just needs to convert it to, well, as we see here as well, converts it to a different form. So that's the terminology. Now, if we go back to the next question, this is talking about the debranching enzyme in glycogenolysis. How does the enzyme address the challenge posed by the branch structures in glycogen molecules? So remember, we spoke about the branching before, and we said that there were advantages: solubility, regulation, compaction, etc., accessibility, and so forth. But by adding those additional branches, it also adds an extra layer of complexity when you want to break it down, because the enzymes are very specific to the glucose in a particular shape, in a particular structure. So when it gets to that branching point, you need an alternate way of acting. So glycogen phosphorylase cannot cleave the alpha-6 1,6 bonding glycogen, so that is a big problem. And it stops phosphorylasis 4 residues away from the branching point. And so if that's where we stopped, then there would be a lot of glucose that would not be acted upon. So what the cell does is that it remodels the glycogen, and it uses a transferase and alpha-6 glucosidase. So these are the 2 enzymes that add to the 4 that we spoke about previously. And then the free glucose is phosphorylated by hexokinase, and this is again a similar reaction that we've seen previously. So by using this approach, the cell is able to modify the alpha-1,6. By remodeling it, converts it to a form that can be accessible and allows for every single glucose molecule to be cleaved and in turn to be released. And obviously that's going to be a very, very efficient process. So if we now go back to the idea of liver and muscle, well, let's start off with the liver. Detail the mechanism by which the liver releases glucose into the bloodstream during glycogenolysis. So remember, this is the key role for the liver. And what is the role of glucose 6-phosphatase in this process? So we know that glycogen degradation has to be controlled, and it's controlled by regulating glycogen phosphorylase, because as we just saw previously, that was the enzyme that cleaved the glycogen into glucose, and it's telling you the same thing here, glycogen degradation. That should make perfect sense. How do we control this enzyme? Well, there are several allosteric effectors. These all act as signals to the energy state of the cell. And again, this is something that we have seen time and time again from a metabolic perspective, that as products are being generated, they feed back or loop back to inhibit the enzyme that has led to their synthesis. So that's one approach. We've also seen control through phosphorylation. And remember, phosphorylation can be reversible. So glucagon, adrenaline, and insulin can all alter the phosphorylation state depending on the requirements of the individual or the cell. So if we look at the liver, we have two forms. There's the active phosphorylated form of phosphorylase. So it's called phosphorylase A. Easy way to remember, A for active. So phosphorylase A is the active form. phosphorylated, and again, that's what you typically see. You put the phosphate and it turns something on. And the inactive form is the unphosphorylated form of phosphorylase B. So you only need to remember one half because you can predict what the other half is going to be. So the liver has mostly the A form, the active form. And this makes sense because the liver is responsible for providing glucose to the whole body, so it's going to need an active enzyme that is able to respond very quickly for the needs throughout the individual organism. When we've got varying amounts though, the binding of glucose to the enzyme changes it to the inactive state. So if you've got excess amounts of glucose being produced, we want to feed back and turn it off. That comes back to this idea up here of allosteric. So glucose is an allosteric modulator of glycogen phosphorylase. So changes it to the inactive state, makes it less active. Since excess glucose is present, there's no need to degrade glycogen. And again, this is the same idea we've seen time and time again, and we'll see in future lectures as well. Once you make enough product, the product goes back and inhibits the enzyme. And that's a very efficient way of doing things. Otherwise, you end up with excess product that is wasteful, but it can also cause other issues throughout the cell. Now, we've got that idea of phosphorylase. So let's talk about the regulation of phosphorylase activity in the liver, but also the muscle, because obviously we've got glycogen being converted to glucose, but we've got the needs of the individual organ, and the organism will vary between the liver and the muscle. So in the muscle, the default state is the off form, and that should make sense because you don't want in the muscle the glycogen being converted to glucose because it needs to be stored when there is exertion by the muscle. So until that occurs, there's no point converting it to glucose. So as it says, only activated during muscle contraction. And this leads to high levels of AMP. And AMP, remember, is the precursor, or can be converted to ATP. So if you've got high levels of AMP, it indicates that the ATP has been converted or been used, so energy has been expended. Glucose 6-phosphate can in turn stabilize the OFF or the B form. And phosphorylation is what converts the inactive, to the active form. Low blood glucose leads to glucagon release, while exercise releases adrenaline, so you can tell the difference in terms of storage versus release, and this leads to phosphorylation and hence activation of the enzyme. So as always, you've got one reaction, you've got an opposing reaction. That is very important for control mechanisms, otherwise you've got no way of actually regulating a pathway or a process. Okay, so now we're talking about degradation, but what about synthesis? So contrast the processes of degradation and synthesis. How does glycogen synthesis use UDP glucose as the activated glucose donor? So glycogen uses uridine diphosphate glucose, so glucose. Uridine, we will talk about in the context of nucleic acid. So we've got uracil, uridine, all very much related types of molecules, but in this case, it's being used from a biosynthetic perspective. So it's used here in favor, or in contrast to glucose 1-phosphate. So this is the activated glucose donor. Remember, when we look at activated, this is essentially telling you that it has phosphates attached, uridine diphosphate, and those additional phosphates are where the energy can be stored and it facilitates the reaction going forward. So if we look at synthesis, you've got your glycogen molecule. Here is your glucose with the activated UDP. The energy is used to then add an extra glucose molecule, so you can see glycogen N+1, and we release the UDP. It's there just as the carrier. The degradation is the reverse. Glycogen, we use UDP and phosphate. That takes away one glucose molecule. We've got glycogen that is now smaller by 1 and glucose 1-phosphate that has been released. And as I mentioned, UDP glucose is the activated form of the glucose, which facilitates this through the energy process. Now, what about the role of glycogen synthase in glycogen synthesis? What is its role, but also the significance of glycogenin in the process? And you'd see glycogenin, when you go back to your very first diagram, there's a yellow G in the middle of the glycogen molecule. That's referring to the glycogenin precursor. So, glucosol units, so this is just glucose, remember glucosol units are added to the non-reducing end of glycogen. And glycogen synthase is that key regulatory enzyme that adds those units. So again, key regulatory enzyme point of control. It takes the glucose from the UDP glucose that we saw previously and adds it to the growing glycogen. Not surprisingly, we've got two forms, liver and muscle. They will differ because the demands of the organ are going to vary. Synthase only adds glucose residues to a polysaccharide with 4 or more residues, and therefore we need a primer. We need something that allows it to begin, and this is where glycogen comes into play. Glycogen. Comes into play. Glycogen acts as the primer. It acts as the starting point so that the glycogen synthase is able to bond to the glycogen, and start to add those glucose residues to it and start to build a glycogen molecule. That's an important process. So we've spoken about the degradation from an energy perspective, but once you have excess glucose and you want to store it, or you need to replenish the stores of glycogen in the muscle, this synthetic process is also going to be important. And lastly, let's talk about the regulatory effects of insulin on glycogen metabolism. Insulin is a hormone, and it has to inactivate glycogen synthase kinase. And it does this through a receptor, which is called a tyrosine kinase. Again, we'll talk about this type of signaling in the signal transduction lecture, but basically what this means is that the receptor that binds to insulin is itself a kinase. The receptor also has kinase activity. And what does it do? It adds a phosphate, but it adds it to tyrosine residues. Okay, that's all it's telling you. So binding of insulin activates the kinase. The kinase phosphorylates a target, and that phosphorylation target is called the insulin receptor substrate, or IRS. Those proteins then mediate downstream effects. So in this case, phosphorylated proteins cause recruitment of GLUT to the cell membrane. This is a glucose transporter. That's what GLUT stands for, glucose transporter. This allows the glucose to be transported into the cell, as you can see that here. The activated protein kinases in turn phosphorylate glycogen synthase kinase, and that facilitates the reaction. And similarly, if we've got a kinase activity, we've got protein phosphatase 1, PP1. So this is a phosphatase that dephosphorylates glycogen synthase and to turn off the cycle. So we've got activation of the glycogen synthase and we've got the deactivation. So again, you can see, depending on the signal, you get a signal transduction. It's called recruitment. Basically, what this means is that the response to insulin is added to tyrosine residue, and that phosphorylation target is called the insulin receptor substrate, or IRS. Those proteins then mediate downstream effects. So in this case, phosphorylated proteins cause recruitment of GLUT to the cell membrane. This is a glucose transporter. That's what GLUT stands for, glucose transporter. This allows the glucose to be transported into the cell, as you can see that here. The activated protein kinases in turn phosphorylate glycogen synthase kinase, and that facilitates the reaction. And similarly, if we've got a kinase activity, we've got protein phosphatase 1, PP1. This is a glucose transporter. That's what GLUT stands for, glucose transporter. This allows the glucose to be transported into the cell, as you can see that here. The activated protein kinases in turn phosphorylate glycogen synthase kinase, and that facilitates the reaction. And similarly, if we've got a kinase activity, we've got protein phosphatase 1, PP1. This is a glucose transporter. That's what GLUT stands for, glucose transporter. This allows the glucose to be transported into the cell, as you can see that here. The activated protein kinases in turn phosphorylate glycogen synthase kinase, and that facilitates the reaction. And similarly, if we've got a kinase activity, we've got protein phosphatase 1, PP1. So this is a phosphatase that dephosphorylates glycogen synthase and to turn off the cycle. So we've got activation of the glycogen synthase and we've got the deactivation. So again, you can see, depending on the signal, you get a signal transduction event. Phosphorylation leads to activation and biological effects. And then if we take away that phosphorylation event, we can reverse the process. And this is what we see here. Okay.